All right, we're live. And hello, everyone. Welcome to Being Patient's live talk. And this is Nicholas Chan, a reporter with Being Patient. And today we'll be talking about cannabis's potential neuroprotective effects with Zhibin Liang, or Ben, who is a Glenn postdoctoral fellow in cellular neurobiology laboratory and the Paul F. Glenn Center for Biology of Aging Research at Salk Institute for Biological Studies. So Ben, thank you for joining us and for taking the time to talk about your research.
Yeah, thanks for having me. It's my pleasure.
Yeah, so let's get right into it then, Ben. Tell us, broadly here before we dive into the details about your recent study is, what do researchers know and don't know about the potential protective effects of cannabis for the brain in context of aging and neurodegenerative diseases?
Okay, yeah, I think it's the medical cannabis research kind of really emerging research field. So yeah, and well known is there's the phytocannabinoids, we call it the compound. It's mostly the cannabinoid from the cannabis plant. So they have well known they have acting on the cannabinoid receptors in the brain or is in the central nervous system or in the peripheral nervous systems. So it's a well known that these kind of compound, especially THC, tetrahydrocannabinol, this the psychoactive compound. So they will have some pharmacological effects. For example, can have a pain, release pain, and then also can affecting some research affecting the cancer, and then also there's can improve the memory or behavior, and also there also some research from the neurological disorder that can benefit for that. That's the well known because it's the classical pathway we know is for the cannabinoid receptor signaling. But actually, is right now most people study is the two compounds, the THC. Another one is the tetrahydrocannabinol, and this the cannabinol is CBD, right? And then there's the two major compound cannabinoids in study, but it's the less study for the minor cannabinoids because it's phytocannabinoids right now has been identified from the plant. There's over 150 cannabinoids. So, but most people in the past decades mostly study is the THC and CBD, but other minor compounds lesser study. So there's a kind of less study and unknown. So yeah, pretty much and in the field. And then for the Alzheimer research, kind of a new area using the cannabis. And then some effects showing is the cannabis plant extract, and also it's a pure compound, the THC or CBD can improve the memory and then can reduce the plaque, amyloid plaque from the Alzheimer's. For example, in the mice study and some study that's in limited clinical trial, but that's and also the study is not comprehensive, honestly. And right now, I know there's some studies doing the clinical trial using CBD, and then to study the effect of CBD for the Alzheimer patients. But there's a study, this class of study for the medical cannabinoids is really in the past couple of years, but it's not comprehensive, I would say that. So it's kind of a new area for the medical cannabis research. Yeah.
So in context of Alzheimer's research, scientists have been looking at cannabis's effects for cognition, for behavior, and for even biomarkers like amyloid plaques.
And the plaque, yeah. So there's a more classical study, and yeah. So, but we also know that is the besides this kind of classical marker, cannabinol also have another interactions with other targets and potential new marker, and then can be potential therapeutics.
Got it. And yeah, before we talk about, your team's focus on CBN, cannabinol, which is different from THC and CBD. Can you tell us, so like, would you say that, the evidence in terms of medical cannabis's effects for Alzheimer's patients, is very early stage right now? And
yes.
that's a very early,
Yeah, it's a very early start. Yeah. Yeah, you go ahead.
Can you give us a sense of like where the research is at in terms of how strong or how weak or how early these evidence are in terms of its effects for Alzheimer, for people living with Alzheimer's?
There's some study in the clinical trial, and then it's ongoing, and then it's you showing the either THC and then potential CBD can clear some amyloid plaque. That's the more classical marker for the Alzheimer's, and also can improve some cognitive functions and memories in I would say that it's a preclinical animal study and also some limited clinical study in human beings. But the study I would say is not the size is not big enough, and then the data is not. I would say that again it's not comprehensive. So it's a more study and then more rigorous evidence need to be shown in the future. So really saying that cannabinoids, THC or CBD or any other cannabinoids really have a beneficial effect. Yeah.
Got it. And just to be clear to our audience, like, there are no cannabis products out there that is FDA approved for Alzheimer's, right?
No. Yeah. There's all in the clinical trial, some of in the clinical trial, but it's not complete. It's very early stage. Yeah.
So yeah, let's get into CBN. Why has your team decided to, focus on CBN rather than THC and CBD when we're studying cannabis in the context of aging, neurodegeneration?
Yeah, that's a good question. There's because when we studied medical cannabis, and then we found there's many compounds, cannabinoids and all the chemical structurally is very similar, but is that THC is the psychoactive compound. So these class of compound, if we're affecting, definitely we're affecting the people's mood, and also is the and the brain's function, and also the behavior, psychological behavior would get changed. And then we're thinking about for the long-term use for the Alzheimer patient, because it's the chronic diseases, right? And then so if for long-term, this THC, this psychoactive compound is not ideal for the patient, because we can predict it's really strong side effect. Yeah, and also CBD, and then that's why people study CBD, because these compound is the non-psychoactive compound. So they also have a beneficial effect. But when we study is that besides these two, what are there any other non-psychoactive phytocannabinoids will be beneficial? And then what is the potential mechanisms for the future benefit for the Alzheimer patients? And that is how we inspire our research. We want to study a minor study of phytocannabinoids. And then later on we focus on the CBN. And what's the CBN? And then maybe we can clarify what's the difference from CBN, THC, and CBD. And then besides is the mechanism of action. And then they structurally have a very similar, but CBN is the degraded products of THC. So basically it's mostly found in the very aged cannabis plant. So if you have a fresh cannabis plant, and then you put it for many years, and then the concentration of CBN will increase, but THC will degrade it. So that is the CBN is the degraded product of THC. But very interesting is that CBN is no longer psychoactive compared to THC. So we think is that this compound could be it's a minor compound, but there's a potential is the new candidates for us to explore the beneficial effects of cannabinoids.
Right.
Yeah.
So that like and down the line, if it could actually be turned into like a product for patients, like long-term use, that would be better because it doesn't have the psychoactive component.
Yeah.
And when we say psychoactive, like to translate to like layman's term for the general audience, does that just mean like THC gets you high? Like
yeah.
Is that?
Yeah, that's something is that they get you high and then get you affecting your decision and your mood and some other part of your daily life. And then so honestly, is for the this kind for Alzheimer and the other neurodegenerative diseases, for the long-term use for the in the patient is not really ideal drug candidate.
Yeah, got it.
Yeah.
So yeah, then tell us about the evidence of so far of your team's research behind CBN and potential.
Yeah, it's happy to share. So basically, is our study rationale is we think is so for the aging, and then it's called aging is one part of them is your brain's energy is the decrease. So the energy production or the energy metabolism really is produced from the mitochondria. So our initial idea is so whether is any compound will be have a benefits and then help for the mitochondrial function, which can help to the Alzheimer patient or other neurodegenerative diseases. As we know that is in the aging process, mitochondria is become in the brain cells it's become dysfunction or is the inefficient, and this one is more dramatic in the neurodegenerative diseases. So these happen to, besides my Alzheimer's, also happen in Parkinson's disease, Huntington disease, frontotemporal dementia, and you name it, many diseases. So is these mitochondrial dysfunction cause many consequences. One is the redox. So it's the cause of oxidative stress increase in the brain cells, and also accumulates many oxidized or dysfunctional proteins and accumulate in the aging brain cells. So that's why it's the aging dependent changes we think is the mitochondria will need to be taken into account to better study the neurodegenerative diseases. So we can understand the root causes of the age-related diseases, and also can ultimately translate the biology of aging into the novel neurotherapeutics. So that's the basic rationale for us. So when we studied that is that we screened I think it's in 2019 we screened a large amount of phytocannabinoids, including THC, CBD, and also CBN and other cannabinoids. We found that all these compounds is affecting one pathway, and then in our cellular model called it oxytocin or ferroptosis. So these I want to mention a little bit is the oxytocin ferroptosis is the non-apoptotic program cell death pathway. So this activation, this pathway can activate the oxidative stress in the brain, in the cell, and this process pathway is recapturing many key features of mitochondrial dysfunction. So we're using this assay to screen a large library of the cannabinoids, and we found is that many cannabinoids, including THC, CBD and CBN also protect the brain cells from deaths against oxytocin and ferroptosis. So, and that's a trigger us very interesting is because we think our original concept is that, okay, it's THC and then affecting CB1 or CB2 cannabinol receptors. But in our model, and then we previously confirmed this cell model for the oxytocin ferroptosis is the no CB1 or CB2 receptor. So what kind of a mechanism is that? So that's why, and then but at least the data showing is that there's the potential novel neuroprotective mechanisms for these phytocannabinoids. So we later on choosing CBN because I mentioned we don't want to study THC because it's a psychoactive compound. And also people, different group in the world study CBD already. So we try to study other compounds. So we choose CBN to do the study. Yeah, so that's and then we found out the key finding is we found out is the compound, the CBN, is directly targeting mitochondria. So the compound definitely is interact with mitochondria, so can reduce the oxidative stress in the brain cell, and also can so I would say is how to have benefit to modulate the energy metabolism, so increase the ATP production into the brain cells. And this one is very interesting, and totally not affecting CB1, CB2 receptors, so independent of this, the classical cannabinol receptor pathway. So that's the major findings for in our study.
Right.
Yeah.
This is the recent study that was published this year that you're
Yeah.
Essentially that's that CBN protected nerve cells from the oxytocin ferroptosis pathway, and like your team wrote, it helped preserve the key functions of the mitochondria, right? That's
that's something we found is because this kind of a normal finding is that at this people more have a perception is CB1, CB2 receptor is the major pathway the cannabinoid we are affecting. So there's a trace this pathway to do the study, but our study cover go to the newer area, so have a mitochondrial function and the cannabinoid, specifically we talk about the CBN is affecting mitochondria. So that is a very interesting part. And then we found that in the oxytocin ferroptosis condition, the mitochondria is more curled up and then kind of rounded and then get very fragmented. So basically, it's get sick or it's the dysfunction for the mitochondria. But when we treating the cell with CBN, and then the mitochondria become more functional, and then it's more elongated and more similar to the control and healthy cell, healthy mitochondria. So that is the key finding for us. And also there's an implication is that because mitochondria is the mitochondrial dysfunction, we say that is not only present in Alzheimer's disease, and then also presented in other neurodegenerative diseases, Huntington and Parkinson and ALS, for example. So and also not just the brain, and also there's in muscle, in heart cells, and other part of the cell. So there's the CBN if kind of independent for the CB1, CB2 receptor, but it's affecting mitochondrial function. That it have a more broader benefits for the other diseases. Yeah.
And what about the next steps for the research then? How are you going to move it forward?
Yeah, that's we are planning, and actually we're doing right now. And then our published paper is more focused on the cellular level, so in the neuron. So now and then we try to testing CBN in the preclinical animal model. So using the mice model for the Alzheimer's disease and aging, we try to reproduce the effects and then see how the CBN actually working in the animal models. And then couple of things we cover more focus. First is the safety, profile, and then the pharmacokinetic profile, and also there's the efficacy, how well this compound will showing the benefit for the aging and Alzheimer's disease mouse model. And then we hopefully, if we can get some positive results, and then we'll share with you later on. Yeah.
And then really down the line, like if it pans out, then maybe test it in humans, right? But that's
that's a long step, because all the compound for the drug development campaign have a long time, and then kind of a step, and also be very rigorous, and then be very careful to really based on the science, right? But there's some in terms of the safety, I would say that is that there are some limited animal study showing this CBN is safe in animal, mostly just in mouse and rats. And also there's some small scale of the human clinical study showing the CBN have a safety profile is reasonable. So that's the kind of optimistic for us. So, but also actually, so we need to test it in a more rigorous, and then hopefully, and then we're showing is the more strong evidence to have making the claim the CBN really is a good candidates for Alzheimer's than other neurodegenerative diseases. Yeah.
So for the Alzheimer's animal models, are you going to be looking at like CBN's effects on like the animals? Like cognition and things like that.
Cognition, memory, so but I think is we're using the classical model, using the amyloid based mouse model, and then so to see that's more classic for the Alzheimer's disease research. And then in our lab we also have using this rapidly aging mouse model, so that is more relevant to the aging process. So we try to compare both of them and then see what's the function will get changed, especially we talk about the mitochondrial function in this animal model. Yeah.
And is this in terms of testing it in animal models, is this ongoing or is this in the future?
It's ongoing. Yes, we're already starting the animal study already. So but it's just the study for in the past month. So that's hopefully is that we can get some good results at the end of this year or this next year, early next year.
I mean, this is really interesting. Like we hear like really the steps in the scientific in the scientific discovery process, in terms of like you start at the cellular level and then you move on into the from the animal models, you see how that goes, and then maybe test it in humans.
So that's a step by step. And then because we in our lab is the cellular neurobiology laboratory, so we more focus on the cellular level first, and then so we will move on from once we find the compound it's really protective and work in the cellular level, and then we advance to the animal study, and then so hopefully, and then to see is the pharmacological effect is the and efficacy, and then working in the animal model, then we will more move on to the potential human clinical study.
Got it. Well, is there any last thoughts that you want to add?
So is I would say that is that cannabinol medical cannabis, I would say that is or medical cannabinoids. The field is really emerging field. So and then so there's a lot of people look at this class of compounds, and then we explore the new beneficial effects. But I would say that is that our advice or is the advocate is that try to make this compound is the more claiming based on the science, not just on the commercial or some other people, and then put it and then not based on the anecdotal results, not really on the scientific base. So we hopefully, and then so it's more and more people working on the field, and then accumulate more data result and evidence to support the medical cannabis and in the future. But so far is I would say that the field is very early, and then everyone and then studying in different area. But at the end, we say that all the result or all the data should be come together and then really make clear a strong evidence to claiming is the cannabinol is really helpful or benefits. Yeah.
Right. But to date, there's no conclusions made.
No conclusion. Yeah, no conclusion is that. But that's how the science work, and then everyone working on their own, and then so individual and then study. But all the data accumulate, and then during the years, then it will be more clear, and then to be more showing the clear results, and then can be support the claim of the cannabinoids. Yeah.
Got it. Yeah, I mean, because there's so many products out there from companies that are, with many.
Yeah. So but there's many issues whether their product is have a pure or how the purity issue, and also it's a claiming not really based on the double-blind clinical trial result, and then or it's really it's maybe just anecdotal. It's just with small scale study, and then so that's how. I mean, it's for the science is not good for that. Yeah, and also for the medical use is that we need to be very, very careful. Yeah.
Got it. Well, thank you so much, Ben, for your time again, and yeah,
it's my pleasure. It's my pleasure. Yeah, thanks for having me and then share my thought. Yeah.
Yeah, and for our audience, if you missed any of this live talk, we'll have a recording uploaded to our YouTube channel, as well as a transcript published on our website. And if you haven't signed up to our newsletter yet, don't forget to do so on our website. Thank you, everyone, for joining. Bye.
Yeah, thank you, everyone. Thank you.